We'll be differentiating between OTOs and ASVs in microbiome analysis. So definitions: first, OTO is operational taxonomic units. These are clusters of amplicon sequences grouped by similarity that is typically greater than or equal to 97%. Proxy for species, but actually a fuzzy operational grouping. ASVs are amplicon sequence variants, inference of the true biological sequences present in the sample after correcting sequencing errors. Single nucleotide resolution, each distinct sequence is equal to ASV. Now, comparison table. So we have the feature OTOs and ASVs. Concept is for OTOs, groups of sequences clustered at similarity threshold, for example, 97%. ASVs exact biological sequences inferred from after denoising. Threshold dependency arbitrary. You can choose 97%, 95%, 98%. Right? There is no threshold. No clustering is done. Right? Resolution species proxy often merges species or strains. ASVs single nucleotide differences retained. Reproducibility study specific OTO boundaries shift with data set size and clustering method. Universal ASVs are universal. ASV sequence is reproducible across studies. Then OTUs are have the errors that get merged into OTUs or inflate counts. ASVs errors modeled explicitly data to deblur and removed. OTUs can work de novo, that is no reference required, while ASVs also are reference free but more robust to missing data. Now OTUs have basically pairwise distance plus clustering, so heavy computational load for big data. But ASVs, since we are using denoising algorithms which are computationally intensive, but it is scalable. Then OTUs assign representative OTU sequences to taxonomy, and ASVs assign each ASV to taxonomy. OTU 97% cluster, loosely defined species-like unit. ASV actual DNA variant, biologically interpretable. Then pipelines, Mother, QIM1 research in OTUs, and QIM2 data to deblur and data to R package in ASVs. Now, use cases: legacy studies, ecological diversity at broad taxonomic levels for OTUs, and for ASVs, modern studies, strain level resolution, reproducibility. Now, why the shift matters? So OTUs were crucial historically, but are study specific, hard to compare across labs. And ASVs are absolute biological units, can be shared across data sets, combined in meta-analysis. ASVs enable strain tracking in clinical or ecological studies. Now, concrete examples. So, example one is E. coli versus Shigella flexneri. OTU 97%, they share greater than 97% 16S identity, group into same OTU. Now, ASV, single base differences preserved. So, separate ASV is possible to distinguish strains. Example 2, mock microbial community, known species mix. OTU pipeline might overestimate, that is splitting same species into multiple OTUs, or underestimate merging close species. And ASV pipeline reproduces exactly the expected number of taxa with accurate relative abundances. Now, the big picture takeaway is OTUs are operational shortcuts, useful when sequencing was noisier. But ASVs are modern standard higher accuracy, reproducibility, and resolution. Today's best practice is using ASV-based methods, data to deblur for new studies, and use OTUs only when comparing against legacy OTU-based data sets. Now, talking about data 2 and the motivation versus OTUs, short concrete. So, the short concrete version, right? So, problem with 97% OTUs is arbitrary threshold. We have set 97%. Loses single entity differences, strain subspecies, non-reproducible clusters that change when you add data. The lecture slides that I uploaded call out exactly these limitations. Data 2's promise was that we'll be inferring the actual biological amplicon sequence variants (ASVs) by modeling sequencing PCR error processes and splitting real sequence variation from the errors, resolving differences of as little as one nucleotide. This produces reproducible exact labels that are ASV sequences that can be merged across studies. Now, the high-level idea is that Data 2 models the sequencing error process, Illumina, and computes for each unique observed sequence the probability that it could have been produced by sequencing error from a more abundant true sequence. If that probability is too low, that is, the sequence is too abundant to be explained by error alone, Data 2 calls it a new true variant ASV, amplicon sequence variant. The algorithm is a divisive, iterative partitioning scheme that ends when all remaining unique sequences are consistent with being produced by sequencing errors from their partition centers. Now, concrete steps mapped to internal Data 2 actions. So first we have dereplication, collapse identical reads into unique sequences with counts and consensus quality profiles. So we work on unique sequences plus abundances. Then initial partition, start with one partition containing all unique sentences. Start with one partition containing all the unique sentences. The most abundant unique sequence is chosen as the initial center of a partition. Then error model, learn the errors. Estimate an Illumina run specific error model. Data 2 learns the per base substitution or transition rates as a function of nucleotide, position, and quality scores, so it knows which substitutions are more likely and how quality affects that. The model gives an expected rate lambda j to I for observing read sequence I given true sequence j. This error model is central. It encodes sequencing biases. Right, error model. Then abundance p-value. So for each unique sequence I in a partition and a candidate center j, compute an abundance p-value. The p-value of observing, or the probability of observing, the p of observing as many or more reads of I if they were only sequencing PCR errors of the center j given the lambda rates and the observed counts. In plain English, since you might not have understood any of this, is the sequence I too frequent to be explained as an error from J? If the Boniferini corrected p-value less than threshold omega A, treat I as evidence of a new true variant, form a new partition centered at I. Omega A controls the sensitivity, default is extremely conservative, 1e minus 40. 1e 40. Now iterative splitting, that is 10 to the power 40. Then iterative splitting and reshuffling. After one new partition is created, the algorithm reshuffles sequences each of the unique sentence that is assigned to the partition whose center most likely produced it. Highest likelihood under the error model. It recomputes the p-values, pick the smallest p again, split further if needed. Repeat until all unique sequences abundance p-values are greater than omega A. That is, each sequence is consistent with being an error from its partition center. This yields the final set of partition centers, that is ASVs. The algorithm therefore is divisive, split, which with the statistical decision rule abundance p-value rather than arbitrary percentage identity cutoff that we used in OTUs. Now moving on, we basically in this chat I have also gotten the practical implementation if you want to go through R or QIM2 examples. And we have the key parameters, what they do and trade-offs. So we can go through this. We have filter and trim, learn errors, data, pool, omega A, which is the sensitivity cutoff for new partition. Default we set as 10 to the power 40, extremely conservative. Increase, make less conservative. Sorry, 10 to the power minus 40. Increase, that is make less conservative to detect the rare variants, but expect more spurious ASVs if you go too permissive. You can set via set data output. Then remove chimera de novo, merge pairs, min overlap, max mismatch. And now we have the chimera detection, merging and taxonomy assignment, extra details, which you can go through, which is not really in our beyond our syllabus, so we can skip this part. So what you get, outputs and how to interpret. So we get a sequence table, that is an ASV table, which is a matrix of samples across the exact ASV sequences. Columns are DNA sequences. Counts are the number of reads assigned, and this replaces the OTU table. Taxonomy table, taxonomy assigned to each ASV via assigned taxonomy with confidence scores if you request them. Read the tracking summary. Data 2 encourages tracking, reads through steps. Input, then filtered, denoised, merged, and non-chimera. Low retention at any step points to problems. Poor quality, wrong trimming, insufficient overlap. Right. Now limitations, pitfalls, and best practice. Pitfalls and how to avoid them. So not removing primers or adapters before data 2. If the primers remain, they add artifactual variation and break the merging. Remove the primers, for example, using cut adapt before filter and trim. Wrong truncated length, failed mergers. If you truncate too aggressively to remove the low quality tails, but leave too little overlap, pairs won't merge. Plan the trunk length to preserve greater than equal to 12 base pairs overlap. That is the default, including primer or amplicon length. Then default omega A, too conservative for rare ASVs. If you expect rare true variants, consider pool is equal to pseudo and or adjust omega A, but check false positive rate carefully. Now pooling trade-off, pool is equal to true finds rare variants across the run but costs time and slightly higher chance of spurious ASVs. Pool is equal to pseudo is intermediate and often recommended when you need extra sensitivity. Then chimera method selection, for very large studies use method is equal to consensus for chimera removal. For smaller datasets, pooled can be more sensitive. Best practice summary now is trim adapters, primers with cut adapt first, then run filter and trim with max EE and sensible trunk length. Visualize fast QC per base quality. Learn errors per run with learn errors function. Don't reuse across different runs. Consider pool is equal to pseudo for more sensitivity, and this is when detecting low abundance ASVs across similar samples. Track read retention after each step. Low retention flags and they require fixing. Quality trimming, primer contamination, wrong settings, etc. Now benchmarks and evidence data 2. We basically can see this. We can skip. Right, this part we have to skip. And yeah, that's it. Now moving on. Yeah, so now I'm moving on. What data 2 is trying to do and how the slides map to that goal. So data 2 basically replaces the heuristic 97% OTU. Heuristic is basically the educated guess that we apply. 97% OTU clustering step by trying to infer the actual biological amplicon sequences that generated the observed reads while explicitly modeling the sequencing error process so that the abundant true sequences are not collapsed with rare error derived reads and rare genuine variants are not discarded as noise. The slides show a simple conceptual pipeline which represents the distinct unique sequences observed in the dataset after collapsing identical reads. Right. So the first concrete computational action data 2 takes is basically dereplication. That is, identical reads across the dataset are collapsed into a single unique sequencing record that stores the DNA sequence, the number of times that exact sequence was observed, that is its abundance or its frequency, and a consensus profile of base quality scores for that unique sequence. Dereplication is important because it reduces redundant computation, right, and preserves the abundance signal that is essential for distinguishing true variants which tend to produce many reads from random errors which typically produce few reads, right. Now, all identical sequences grouped into clusters of unique sequences. Each unique sequence is now a data point with count and quality information that the algorithm will use. Then step 2 is initialization step, where data 2 places all unique sequences into a single partition or a single cluster. This means the algorithm starts conservatively, assuming for the moment there may be only one true sequence and all the other observed sequences might be errors derived from it. The partitioning approach is divisive. It will split the internal partition only when the evidence, that is the statistical evidence derived from the error model, indicates that some observed sequence cannot reasonably be explained as an error of the current partition center. Starting from one partition avoids the arbitrary ordering biases of greedy centroid clustering and provides a clear statistical rule for when to split. Now, in the slide, I just mentioned all unique sequences are placed into one single initial partition, which is the foundation for split by evidence approach data 2 uses. Step 3 is simple but critical choice of a partition center, where within the initial and later each partition, the most abundant unique sequence is chosen as the partition center, the candidate true sequence. Abundance is used as a heuristic because under a reasonable biological plus sequencing process, a true biological sequence will typically produce many reads, while sequences arising only from sequencing error of to some true neighbor will appear much less often. This center is the sequence from which data 2 will test whether other unique sequences could plausibly have been generated by error. The most abundant sequence is taken as the center of the partition. The choice is the anchor for the entire subsequent statistical test. Now, the statistical error model that we have, now this goes in mathematics, which I've already covered in my previous video, so you can go through that. And I will also attach this in the description so you can go through it and read it. Then we have the abundance p values, again, the mathematical part. And because the algorithm performs this test, let me continue from here. Because the algorithm performs this test that we have done mathematically above for many sequences within a partition, data 2 applies a multiple testing correction, that is Bonferroni, to the set of abundance p values in the partition before making split decisions. The Bonferroni step ensures that the algorithm does not call many spurious partitions just by chance in large partitions. It intentionally biases towards the conservatism. After p-value correction, data 2 selects the sequence with the smallest corrected abundance p-value. If that p-value falls below a user controllable threshold called omega a, that is omega a, the sequence is considered too abundant to be an error and is promoted to its own partition center. Now these steps are called out: compute abundance p-values, Bonferroni correction, pick smallest p less than omega a, and make that a new partition. In practice, the default omega a is set extremely conservatively, that is very low. The package default is very small, that is 10 to the power minus 40. 1e minus 40 means that, which is intended to minimize the false positive ASV calls. Now the users, that is we, can relax this when we explicitly want higher sensitivity for very rare variants, but then we must accept a greater false positive risk as well. Now, after the new partition center is created, the algorithm recompares that center to all remaining sequences in the original bulk partition. Sequences are re-evaluated relative to each center, old and new, to determine which center is the more likely source under the error model. This is not a one-shot split. The algorithm allows sequences to move between partitions if the likelihood under a different center is higher. Now, basically, the remaining sequences are reassigned. Illustrates the re-evaluation step. Concretely, if a given observed sequence I is more probable as an error of the new center than of the old one, it will be moved into the new partition. This keeps the partitioning aligned with the statistical model rather than letting any early split produce stranded sequences in an inappropriate cluster. Then finally, we have steps 8 and 9, reshuffling, iteration, and convergence, why the algorithm terminates with ASVs. So after creating any new partition, data 2 performs a reshuffling step. Every unique sequence is assigned to the partition whose center most likely generated it, according to the error model and the observed abundances. This is actually a likelihood-based assignment, not a simple nearest neighbor by sequence identity. Once sequences are reassigned, the algorithm repeats the split test process. It recomputes the abundance p-values within each partition, applies the Bonferroni correction, selects the current minimal p-value and tests against omega a, splits if warranted, and reshuffles again. This iterative cycle identifies the most statistically extreme candidate, split, then reshuffle, continues until the stopping criterion is reached, which is when no unique sequence in any partition has a corrected abundance p-value less than omega a. At that point, every observed unique sequence is consistent under the error model with being explained as an error of its partition center. Therefore, the partition center are accepted as the inferred true sample sequences. Those centers are the final amplicon sequence variants, ASVs. That's about ASVs, and basically what an ASV is practically and how data 2 outputs it for downstream use. So each partition in the end, each partition center surviving the iterative divisions corresponds to an exact nucleotide sequence, the ASV, which data 2 reports as a canonical sequence string. For example, AC, GT, GCT, together with sample level counts, how many denoised reads map to the ASV in each sample. So the package constructs an sequence tables, samples cross ASVs, analogous to old OTU tables but with exact sequence keys instead of clustered OTU IDs. Because ASVs are exact DNA strings, they are comparable across runs and projects if the same region and trimming strategy are used, enabling meta-analysis without having to recluster anything. So basically, the what ASVs allow is a single base resolution where OTUs lose that signal. Now modified Needleman-Wunsch alignment step and the speed heuristics, k-distance cutoff and band size, what they are and why Data 2 uses them. So after Data 2 infers ASVs, most users want to attach the taxonomy. So Data 2's assigned taxonomy and the package alignment code use a modified and optimized Needleman-Wunsch alignment NW alignment routine to compare the ASV sequences against a large reference. For example, the Silva ribosome RNA database, rRNA database. Doing a full global alignment against every reference sequence would be computationally expensive. Therefore, Data 2 applies two practical heuristics that are in the slides, k-distance cutoff and the band size. So k-distance cutoff is basically k-mer distance pre-screen. Before attempting a full alignment between query ASV and a reference sequence, Data 2 computes a fast k-mer similarity, that is a short k-mer identity metric, and if that k-mer distance exceeds the k-distance cutoff, the code assumes the pair are too dissimilar, for example greater than 10% nucleotide mismatch, to be linked by sequencing error and skips the costly alignment. The default k-distance cutoff was chosen empirically on Illumina 16S data to avoid spending time on pairs that differ by more than around 10% because such differences cannot plausibly be explained as sequencing error of a single template read. In practice, the package default k-distance cutoff is around 0.42, a k-mer distance metric calibrated to approximately greater than 10% mismatch. Documentation and function help note this default and advise re-examination when applying Data 2 to markers with very different evolutionary properties. For example, indel-rich ITS regions. Skipping distinct pairs substantially improves the speed without materially affecting correct classification for typical rRNA markers. Then next we have the band size parameter, which controls the width of the alignment band in dynamic programming Needleman-Wunsch alignment. The algorithm restricts the allowed net number of gaps between query and a reference to the band size window around the diagonal, which makes the alignment faster and uses much less memory because it avoids full matrix calculation. The biological rationale is that ribosomal RNA gene segments used as amplicons are typically gap poor relative to highly indel-prone markers. Therefore, a narrow band is a safe heuristic that accelerates alignment. If you work with amplicons or markers that have many legitimate indels, for example, some ITS datasets, you should increase band size for or disable the heuristic. Otherwise, the banded alignment could miss legitimate alignments and lead to lower taxonomic recall. Right. So that's about that. And